Hello everyone, and welcome to part 2 of the Carbs and Fats lecture. We're now getting into fats or lipids, which are covered a bit in chapters 4 and 7 of the textbook. I didn't love how they were embedded in those chapters. I kind of wanted to pull them out and put them in their own unit of macronutrients, carbs, fats, and proteins. So that's where this lecture material is coming from, chapters 4 and 7. So lipids are a class of chemicals that are not dissolved or not soluble in water. So by definition, they are all nonpolar. We tend to refer to solid lipids as fats and liquid lipids as oils. I will use all three terms interchangeably, lipids, fats, and oils, probably in this chapter, but that's the difference between those three terms. So lipids can be found in our diet in lots of foods that we eat. Anything from animals contains fat, so usually solid fats like butter or lard, bacon or meat fat. Milk contains fat, which is also found in dairy foods, etc. And then here's a nice image of showing you some oil floating on water. Is that probably just poured in? These little droplets down here are probably on their way up to this oil layer because they will not dissolve in water, they will stay separate. So the different important lipids in biology, there's three different types that we're going to talk about: triglycerides, phospholipids, and sterols. So similar to carbohydrates, carbohydrate is a large family of molecules, and then we discussed different classes of carbohydrates: monosaccharides, disaccharides, polysaccharides, and some important examples of each. Same thing here. Lipids is a large class of molecules, and we're going to talk about some of these different classes, sort of break it down. So the most abundant class of lipids or type of lipid that's in our diet and in our bodies are the triglycerides. So triglycerides, you will see, have this basic structure of, they have a head part made out of glycerol and 3 fatty acid tails. Phospholipids, actually, although it's pictured very differently here, it's actually very similar in structure to the triglyceride. It has a glycerol head, and it has 2 fatty acid tails, but then it has this big phosphate group sticking off of it instead of a 3rd fatty acid tail. And then sterols, as in cholesterol, has a different structure. So sterols have this 4-ring structure, so 3 hexanes and a pentane ring. It looks kind of like a honeycomb, especially when it's colored in orange like this, and then has different groups sticking off of it. So it looks very different in structure to triglycerides and phospholipids because there's no fatty acid tails, but it is still a lipid because it's still very nonpolar and not soluble in water. So these are the 3 different classes of lipids we're going to talk about. So first, let's start with the triglycerides. Tri means 3. That's referring to the 3 fatty acid chains. And the glyceride refers to the glycerol, which is a chemical group or a molecule that these fatty acids are attached to. So we can see their molecular structure here. This is the molecular structure of glycerol. It's basically a 3-carbon chain with 3 alcohols coming off of it. And then the fatty acids. So fatty acids are long chains of carbons and hydrogens with a carboxylic acid group at the end. So we take those 3 fatty acids, we pop them onto the glycerol, and we now have a lipid molecule, a triglyceride. This process of taking a glycerol and 3 fatty acids and sticking them on there is taking 4 different things and putting them together. And in the process, we lose hydrogens and oxygens that form water. So again, just like we saw with carbohydrates, when you have monosaccharides forming disaccharides, all right, it's a condensation reaction. This too is a condensation reaction. Condensation reactions are synthesis reactions. So you'll find a theme in this unit is that to build our important biomolecules, carbohydrates, lipids, and proteins, we use condensation reactions, or it involves condensation reactions. So another thing to note about this picture is that a lot of times when we cartoon draw triglycerides, these three fatty acid chains all look the same. So you think of them as being three identical chains, but they're not. Those three fatty acids can be different fatty acids. So you can see that here in this picture. We've got one that's saturated, just carbon-carbon single bonds, and then two that are unsaturated, one that has one double bond and another that has two double bonds. So they're three different fatty acids. So I use those terms saturated and unsaturated. We've come across those terms before in this class when we were talking about alkanes and alkenes and alkynes, so our hydrocarbons. So a saturated hydrocarbon is one that has the maximal number of hydrogens bonded to it, so it's all carbon single bonds. Whereas unsaturated hydrocarbons have at least one double bond. So fatty acids are just long chains. They're either alkanes or alkenes with a carboxylic acid at the end. And the saturated ones have all single bonds. The unsaturated ones have at least one double bond. So you can have a monounsaturated fatty acid with one double bond, or a polyunsaturated fatty acid. So in this picture, for example, we have a saturated fatty acid, a monounsaturated fatty acid, and a polyunsaturated fatty acid. We're also going to talk about trans fatty acids, which technically are unsaturated because they have double bonds. But when we think about unsaturated fats nutritionally, unsaturated fats are actually good fats for us nutritionally to ingest, the ones with double bonds. Trans fats are artificially made, and we'll see the difference chemically in the double bonds that trans fats have that make them so bad for us. So I hate to even associate them with the word unsaturated, because that has a word that has sort of a healthy connotation in diet. But technically what it means, it's a chemistry term that means that it has the presence of at least one double bond. So this is just another image kind of showing you the differences. So saturated fatty acids have all carbon-carbon single bonds, and a monounsaturated fat that has one double bond right here, and a polyunsaturated fat, in this case, that has three double bonds. So saturated fatty acids are just saturated hydrocarbons with a carboxylic acid at the end. And here's a table from the book showing you some common saturated fatty acids that are found in our diet, like myristic acid, which is found in nutmeg, and palmitic acid, which is found in palm oil, and stearic acid, which is found in animal fat. So these are pretty long molecules. Very nonpolar, very long chains of hydrocarbons there. Unsaturated fatty acids have at least one carbon-carbon double bond. And so it has an alkene as a long tail. So it's an alkene with a carboxylic acid at the end. That double bond makes the molecule have this sort of kink, this bend. So when the carbon chain is all single bonds, it's very straight, or it's a zigzag, a straight zigzag molecule. When there's the presence of a double bond, it causes the chain to actually curve and form this U shape or a kink, and that is going to affect the chemistry of our fatty acids. So we mentioned this before, but I'll remind you, when it comes to nonpolar molecules, the only attractive force they have that holds them together is London forces, right? And London forces are greater the bigger the molecule is, but also the straighter it is. So saturated fatty acids, which have these long straight chains, they can pack really tightly together and have more London forces. So they have a higher boiling point and a higher melting point, and tend to be solids at room temperature. Solids that still probably have a fairly low melting point, like butter, is solid at room temperature, but actually if the room gets a little bit warm, it gets pretty melty, because the London forces are still weak types of forces, right? But Foods that contain a high amount of unsaturated fats, of fatty acids that have those kinks and are bent, okay, they can't pack as tightly together. And so they have fewer London forces, and therefore they have lower melting point and boiling point, which is why oils are liquid at room temperature. So sometimes you can just tell by looking at a fat whether it's higher in saturated fatty acids or unsaturated fatty acids by its melting point or its state at room temperature. So we typically think of foods high in saturated fats, things that come from animals, animal fats, and foods that are high in unsaturated fat, oils, usually come from plants. But something that is sort of an anomaly is coconut oil. If you've ever worked with coconut oil or bought coconut oil or cooked with it, it's not actually a liquid at room temperature, it's a solid. Coconut oil, although it's a plant, from a plant, is actually very high in saturated fats, and that's why it is a solid at room temperature and not an oil, even though it's called coconut oil. All right, so this is just a picture of some unsaturated fatty acids, and I'm going to teach you a little bit of the chemistry designations for these. So you'll see this sort of code system, this carbon designation. So it's two numbers separated by a colon within brackets, and then a comma and this funky looking W, which is a lowercase omega, Greek letter omega, dash number. Okay, so the numbers inside of the brackets, the first one indicates the number of carbons in that chain, in that fatty acid. The second number indicates the number of double bonds. So if this was a saturated fatty acid, that number would be 0, and we wouldn't really need this carbon designation. So for unsaturated, so a monounsaturated fatty acid would have a 1 for one double bond. Polyunsaturated fatty acids have 2 or 3. I don't know if there are any that have 4 double bonds. And then that omega number tells us where those double bonds start. So the omega carbon is the last carbon in the fatty acid chain. So the alpha carbon is over here where the functional group is, and the omega carbon is the tail end. So if we have an omega-6 fatty acid, it means the first double bond appears 6 carbons away from the tail. So if we count that out, we've got 1, 2, 3, 4, 5, 6. So the 6th carbon from the tail is where we see this double bond. And then the double bonds occur every 3 carbons. So that's what that designation means. So I could ask you to draw a fatty acid and just give you the carbon designation, ask you to draw it, or I could give you the carbon designation and ask you how many carbons are there, or how many double bonds are there, or how many carbons away from the tail is the first double bond. So just know what that designation means. Or I might give you a fatty acid and then ask you to give me the carbon designation. So can you count the number of carbons in the tail and count the number of double bonds, etc. So I have a question on this slide that says, are these double bonds cis or trans? So remember, cis double bonds are where you have groups that are on the same side of the double bond, and trans is when they're on opposite sides of the double bond. And so if we look at these, we'll just look at this last one down here. If I take my pen and I draw this sort of imaginary line through these bonds, all right, then the groups are anything like this is a group, and then this is a group, all of this is a group, right? All right, so both of these, so for this double bond here on the right, both of the groups are above. In all of these, that's the case. All of the groups are above on the same side of the double bond. So these are all cis double bonds. As it turns out, in nature, when enzymes generate unsaturated fatty acids, they naturally generate and favor the cis bond. And I'll come back to that in a second. So let's do the sample problem first. Draw the skeletal structure of myristic acid, a saturated fatty acid with 14 carbons. So we just want to draw a chain of 14 carbons. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Just going to double count. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Check. All right, so I have 14 carbons, and now at one of the ends, so either the right side or the left side, doesn't matter, I'm going to do it on the right side, I need to add a double bond to oxygen. That last carbon is double bonded to oxygen and single bonded to OH. That makes a carboxylic acid functional group. So the thing to be careful about is making sure you're not adding another carbon, that you're just adding that double bond to the oxygen and the alcohol group coming off of that last carbon. And the next question, it says, is the following fatty acid saturated, monounsaturated, or polyunsaturated? Hopefully you answered polyunsaturated. How many double bonds does it have? The correct answer is 4. This is the number that tells us how many double bonds it has. It has 20 carbons, there are 4 double bonds, and they start 6 carbons away from the end. So you may have heard nutritionally that omega-3 fatty acids are really good for you. Omega-3 and omega-6 fatty acids are both healthy. These are actually essential fatty acids that our body needs to make compounds that we can't make without them. We have to get them from our diet. We cannot generate omega-3 and omega-6 fatty acids. We tend to get a lot of omega-6 fatty acids in our Western diet, and Western diets tend to be fairly low in omega-3 fatty acids. So you see a lot of advertisements for omega-3 supplements, or a lot of foods that are fortified with omega-3, or just, brag about how much omega-3 fatty acids they have, because it is something that the American diet is lacking in, but is important and is nutritious. But that's what omega-3 means. When we talk about omega-3 fatty acids, we're talking about ones that have a double bond, 3 carbons from the end. Alrighty, so we were talking about how all of these double bonds are cis double bonds, that in nature generates cis double bonds in these fatty acids, right? Well, a few years back, a few decades back actually, I think it was like in the 50s, a health study was done, and it was determined that the American diet was leading to an increase in cardiovascular disease. And it was either carbs or fats were the problem. And ultimately, the doctors decided on fats. It made sense logically, because cardiovascular disease, a big part of it is the buildup of cholesterol in the blood vessels, these fatty plaques. And so they assumed that then excess fat in the diet, and there's definitely a correlation there, was contributing to that. Actually turns out they were wrong. Carbs are way worse for us than fats are. But at the time, they thought they were right. Fats are bad, especially animal fats, saturated fats, and cholesterol, which also comes from animals. And so they tried to make a healthier kind of fat. So butter and lard were things that were used heavily in baking and cooking and, a spread on your breads. And so they tried to make a healthier version of it. And they did this by taking oil, which we thought was, which we think of as healthier because it has all these unsaturated fats. And they manipulated those fats. They did a process of hydrogenation, where they added hydrogen and then removed hydrogen, and they turned those cis unsaturated fats into, trans unsaturated fats. And the trans double bonds resulted in a straight molecule that behaves very much like saturated fats, so it packs tightly. And the reason they did this is because people don't like spreading oil on their toast. They want to spread butter on their toast. Butter and oil have different flavors, different tastes, different textures. And so replacing saturated fat with unsaturated oils was just not always something that was doable, that led to desirable texture of foods. So they took those unsaturated fats, chemically modified them to make them behave more like saturated fats, but assumed that they'd still be healthy for you because they were still technically unsaturated. And we call these trans fats because they literally are the trans chemical form. They have a trans double bond. And so trans fats, you've probably heard of, are very, very bad for our health, as it turns out. So the original concoction of margarine was basically a tub of trans fat that was marketed as heart healthy, when in fact it actually will kill you faster than regular butter will. So it turns out that trans fats are not processed properly by our bodies. We have evolved to use cis fats, and it turns out the trans fats we don't utilize very well, and actually they lead to inflammation that contributes even more to cardiovascular disease. So how do you know if food has trans fats in it? Well, one, you can look on the nutrition label here. All right, it might list that there are 0 grams of trans fat. So that's a good sign. But here's the thing about nutrition labels. If there's less than half a gram of something, the food company can round down to 0. So 0 grams of trans fat might actually mean 0.0 or 0.4 grams of trans fat. So how do you know if there really is indeed 0 grams? Well, you want to look at the ingredients. So remember, trans fats are made by hydrogenating oil, by a process of hydrogenation of unsaturated oils. So looking for ingredients that include hydrogenated oils, partially hydrogenated vegetable oil, hydrogenated oils, that's basically secret code for trans fats. So do try to avoid foods that contain those. They have been more or less outlawed by the FDA, so many food companies have removed them entirely from their foods, but some do still sneak them in, and just at small amounts so that they can still say there are 0 grams of trans fats. The foods that you find trans fats in, margarine used to be a big source, now less so. Fried foods, fast foods, snack foods like snack cakes or frosting, snack like popcorn, like flavored popcorns. So again, checking the nutrition facts label because not all brands are the same. The cis fats, or the cis unsaturated fats, are unsaturated fats. These are typically healthy for us, and you can find them in plant oils, so like olive oil. And saturated fats are going to be ones that are sort of, you want to consume in moderation. And these are going to be things that you're going to find in animal foods, animal fats. So bacon and eggs would both be sources of saturated fat. So those are the triglycerides. So again, these are all different types of fatty acids that make up our triglycerides, the sort of major group of fats in our diet. Another important type of fat that contains fatty acids are these phospholipids. So phospholipids are they're generated by cells and used to make cell membranes. So a phospholipid has a glycerol group, and it has 2 fatty acid tails. Some might be saturated, some unsaturated. And then instead of a 3rd fatty acid tail, that 3rd carbon of the glycerol is attached to a phosphate group. And phosphate groups are charged. They're negatively charged. Charged things can dissolve in water. So this phosphate group makes these phospholipids slightly soluble. In fact, it makes the head of the molecule very soluble in water, or in other words, hydrophilic. It likes water, it's attracted to water. But those nonpolar carbon-hydrogen tails are still not soluble in water. So we say they're afraid of water, they're hydrophobic. And remember, anytime we have a molecule that is part hydrophilic and part hydrophobic, or part polar and part nonpolar, we call it amphipathic. So an amphipathic molecule is both polar and nonpolar. And so phospholipids can form these really interesting structures in an aqueous, watery environment. They can form a bilayer, which is what's pictured here, and this is what our cell membranes are all composed of. They're basically giant spheres composed of a phospholipid bilayer, where the head groups are on the outside and inside, where the water is, and then the tail groups are sort of sandwiched between them. So they're hiding from the water because these are the nonpolar parts. Another structure that they can make is they can make micelles. We saw this with soap molecules. Soap molecules do the same thing. There's a lot of important lipids that form micelles in our body and help us to transport fats. So we can have these micelles that we call lipoproteins because they're made up of phospholipids, and in purple here we've got proteins embedded in them, which just make it a little bit more soluble in water. And then on the inside, where those nonpolar hydrophobic tails are, that's where all of the triglycerides and cholesterol that we eat or that we need to transport through our body can travel in there. Where it's comfortable, it can interact with these nonpolar tails. All right, and then the surface of that lipoprotein is just made up of all hydrophilic head groups. So it allows us to transport fats and fat-soluble vitamins, other nonpolar substances through the body. It can also help us to break up fats into smaller droplets. We'll talk about one of the roles of cholesterol as an emulsifier, something that helps us to dissolve fat in water. So the third class of lipids that's important for biology is the steroids. The main one that we focus on is cholesterol. So sterols or steroids are molecules that contain this ring structure. It's a 4-ring structure. It's called a steroid nucleus. And this is the structure of cholesterol. So you can see that steroid nucleus, and then there's some other, a big alkane group over here, and some other little methyl group, a hydrogen sticking off there. So cholesterol is actually a very important molecule that has a bad reputation. So cholesterol is an essential element, or not an element, an essential compound that your body needs to live. Your cells need it. It's an important structure, part of cell membranes, helps to increase the fluidity or flexibility of our cell membranes. Cholesterol is important for generating vitamin D. It's important for the generation of several hormones. Nope, I don't have it in this lecture. But we produce all of the sex hormones, so testosterone, estrogen, they have stero in them as part of their name because they come from they're steroid derived. Another important molecule that comes from cholesterol is bile. And bile salts, which is a modified form of cholesterol here, all right, is important in digestion because when we eat fatty foods, the fat in our stomach and intestines would just kind of glob all together. It would not dissolve well in the aqueous solution that is our stomach acid. And so what bile does, the liver makes bile and it stores it in the gallbladder. The gallbladder squirts it into the small intestine after we eat something fatty, and it helps to break up those fat globs into smaller fat droplets. And then it's just easier for enzymes to get in there and to actually chemically break down the fats. So bile is an emulsifier. It breaks the fat up into smaller droplets and allows it to dissolve somewhat into the aqueous solution of our digestive juices. So like I said, the liver produces cholesterol, but we also get cholesterol from food, from animal foods. And so most people have healthy cholesterol regulation, and if they are consuming a lot of cholesterol in food, then their liver just produces less cholesterol. So the average person doesn't actually have to worry about their cholesterol consumption because their body will regulate, but some people's liver just keeps producing cholesterol even if they're eating a lot, and then they end up with very high levels of blood cholesterol. Those are the people who usually need to watch their cholesterol levels. So one of the important roles of cholesterol here, another one besides making bile acid, is making the cell membranes more fluid, or actually more rigid. So if you have if you want to have a very fluid, flexible cell membrane, then you're going to use several unsaturated fatty acids there because they don't pack as tightly. And so unsaturated fatty acids make for a very more fluid cell membrane, whereas saturated fatty acids will make for a less fluid, a stiffer, more rigid cell membrane. And cholesterol will make it even more rigid than that. Nope, sorry, take it back. Cholesterol reduces the membrane fluidity at higher temperatures, so like normal, like room temperature, but for something like at very low temperature, it prevents the cell from freezing. So it keeps it fluid even at lower temperatures. So it modifies the fluidity of the cell membrane. All right, so just to sort of tie these two together, carbohydrates and fats, the thing that they have in common is that they're both really important energy sources for living organisms. And we'll see how they contribute to energy in chapter 12 when we talk about metabolizing these macronutrients, carbs and fats, and to some extent proteins. But just to sort of big picture it, all right, let's say you eat a little Debbie cupcake, all right, and which is going to be a source of a lot of fat in the form of triglycerides and also a lot of sugar, so you'll get glucose and fructose because it contains a lot of sucrose, which is a disaccharide. I should have pictured that here, but ultimately we will break it down into the monosaccharides. So these triglycerides and monosaccharides will ultimately be absorbed and go to our liver, from the intestines to the liver. We can store some glycogen, some of our carbohydrates we can store in the liver, that's our liver glycogen. But the liver, mostly its job is to send these nutrients to other parts of the body, either for storage or for processing. It can send glucose and fat to the muscle tissues. They need a lot of energy. So we do send a lot of carbohydrates and lipids there. The muscles can store carbohydrates and to some extent lipids as well for energy. Your muscles use a lot of energy when they're active, so that makes sense that they would have some energy storage there. Notice that you have about twice as much energy in the form of triglycerides, fat, than you do in glycogen, carbohydrates, in the muscle. And then in addition to what we can store in the liver and muscles for energy, we have an entire tissue that's designed for energy storage, and that's our adipose tissue, our fat cells. And their sole job really is to store fat, to store dietary lipids that we don't need for energy right now, to store it for later. So a huge amount of our energy is stored in the form of fat. We can store glucose for energy reserves. That's glycogen, and it's found in the liver and the muscles. But if you look at the amount of calories that we can store in glycogen versus the amount of calories we store in fat, we store way more in fat. Fats also happen to produce more energy per gram. So that's why they're such a good long-term storage solution for energy. So that is the end of the carbs and fats section of this unit. Next up is proteins.